So now that we have talked about viruses, let's get to prokaryotes. Let's get to the living things, get to the nitty-gritty of biology and life. So today we're going to be talking about prokaryotes, which are within the domain bacteria and the domain Archaea. So here's a look at where we are on a scale level of things. Last week when we were talking about viruses right here, they were in the 10 to 100 nanometers for their size. And remember, we said we couldn't see those with light microscopes because light waves are larger than that. So this time we are focusing right here, which is where our bacteria and what they sometimes call archaea bacteria, those fall between 1 and 10 microns. So these we are able to see with light microscopes. So what are some of the characteristics of prokaryotic cells? First of all, like we talked about, they are in the domains bacteria and archaea. Prokaryote, that word itself means before, that pro part, before the nucleus. So prior to a nuclei organelle. They are single-celled organisms and they lack any membrane-bound nucleus. And then how they acquire energy varies a whole lot. So they could be autotrophs or they could be heterotrophs, parasites, mutualistic. So that's a huge variety there. So let's take just a moment to look at the structure of the typical prokaryotic cell. Kind of falls into three different categories for what the cell is made up of. The first is the cell envelope, and that envelope contains the plasma membrane, which regulates what comes in and out of the cell. And since there are no organelles, there are invaginations in the plasma membrane where processes such as respiration or photosynthesis occur. And then there's a cell wall, and that provides rigidity, some stability, and is especially important with fluctuation of water pressure within the cell, so it provides that stability. And then some bacteria, or some prokaryotic cells, but not all, have an additional layer called a glycocalyx. Now, that glycocalyx can be very well organized, such as in a capsule, or it can be very loose, and then that's what we think of as a slime layer. So that's the cell envelope. The cytoplasm, of course, contains ribosomes, because we know that every living thing contains ribosomes. Thylakoids, which are specific to cyanobacteria, and then a nucleoid. And that nucleoid is basically a concentrated area of DNA. Remember, it doesn't have a proper nucleus. And then the third category is the appendages. It can have a flagella, a conjugation pilus, or a fimbriae. So this is a closer look at the cytoplasm. So like I said, it contains ribosomes, that's for protein production, and then contains a single nucleoid, where it has concentrated DNA. So if you look at our cell right here, that would be this right here. Interestingly enough, that DNA is a single chromosome tightly wound together. So you can see here in our cell on the right, if you extend it out, that's that single chromosome, all tightly wound together to produce that nucleoid. They also have plastids, which are extra rings of DNA, and we'll talk a little bit more about that, but here they are right here. And then the other thing is that if it's a cyanobacteria, it has thylakoids. And we're going to talk about that a little bit later because we want to discuss how cyanobacteria is thought to have played a huge role in Earth's history. So for now, just know that thylakoids are stacks of discs used for photosynthesis, and those are present in autotrophic prokaryotes, particularly in cyanobacteria. So here is a picture of those plasmids that we talked about. Those are just accessory DNAs, and they're usually circular and very small, and they just contain extra genes. And sometimes this is where the genes are located for antibiotic resistance, so they do play an important role. So as we discussed, there are three types of appendages associated with prokaryotic cells. There is the flagella, which is towards the one end, and as it circles around in 360 directions, it propels the bacteria forward, much like a propeller would work. The fimbriae are these small hairs on sort of throughout the sides, and they usually help with attachment to a host if it is a parasitic bacteria. And then there's the conjugation pilus. That is actually not in this picture. We will go on to the next slide to show you what that is. So here we go. On this one, here is our conjugation pili, pili being more than one. But if you notice, the conjugation pili comes from the cytoplasm and out. And what that is used for is to allow for transfer of DNA from one bacteria to another. So that's what that's for. And again, you can see the fimbria here and our flagellum here. So those are three types of appendages that we see on a prokaryotic cell. So since we mentioned these appendages, let's talk about how prokaryotic cells move. Most of them do move using the flagella appendage. So a flagella is composed of a protein called flagellin. It's wound tightly in a helix and anchored into the cell by a basal body. Sometimes that's also called a kinetosome. And that basal body is made up of microtubule structures near the cell surface. And it, like I said, it allows that flagella to rotate in 360 degrees and allows the bacteria to move forward just like a boat propeller. It's important to note what I have here in blue. Prokaryotic cells do not have cilia, so don't confuse the fimbriae that we looked at before with cilia. Only eukaryotic cells have cilia, and they use those for motility, but prokaryotes only have flagella, not cilia. So here's just a close-up look at the flagella of a particular bacteria, just so you can see where it all attaches. So here's the glycocalyx on the outside, the cell wall, and then the plasma membrane. And so the anchor, really, the basal body, does really go all the way through into the membrane and allows for that flagella to rotate. Talked in generalities about flagella. There's a difference with our flagella within the domain bacteria and the domain archaea. Remember, they're both prokaryotic cells. The flagella in bacteria is what you have pictured on this slide. It's thick and the basal body is present. So the basal body, if you look at these micrographs over here on the right, this is a cross section through one. Each of these little sections here are made up of a triplet of microtubules, and there are 9 of those. And that's very specific to bacteria basal bodies. That's how the flagella is attached. The prokaryotes in Archaea, the flagella is very thin, and they actually lack a basal body. Instead of having a basal body, there is a flagellin hook that attaches the flagella. So that's the difference between those two domains. So how do prokaryotes reproduce? Their reproduction is asexual, and it's through binary fission. So prokaryotes are haploid, meaning they only contain one copy of the chromosome. So that is specific to prokaryotic cells. So let's talk a little bit about this binary fission. Binary fission is literally the splitting of the cell into two and all of its contents into two. So the way that works, let me get my little pointer here. All right. So first of all, the cell has to replicate its DNA. Its nucleoid creates a second copy of the DNA. And then the cytoplasm fills up the space of an elongated membrane. And then as we continue to go down, cross walls form. And then those cross walls are complete, which separates the cell into two, and then therefore you have two daughter cells. So like I said, just a very simple way of reproduction. It's asexual. It's called binary fission. Okay, so since we only reproduce asexually, how does recombination occur? Remember, recombination is what allows for the introduction of new genes and the possibility to adapt to your environment. Well, this happens in three different ways. The first being conjugation, and that's where the pilus appendage that we talked about comes into play. So remember, it comes straight out from the cytoplasm and through the envelope. So it is literally, DNA is donated from the donor cell all the way to the recipient cell. And so a sharing of DNA occurs that way. That's conjugation. The second way is transformation. Transformation is when the cell picks up free pieces of DNA either from living cells that have secreted it or from dead cells as they break down and their DNA is released into the environment. Then these bacteria can pick it up. The third way is transduction, and that's when the bacteriophage carries portions of the DNA from one bacteria to another. So remember, the bacteriophage inserts viral DNA into the bacterial DNA, and when it's clipped out, a piece of the bacterial DNA may be clipped with it, and then it's transferred into new bacteria, so that allows for the new possibilities of genes. So now that you know some characteristics of the prokaryotic cells, let's just look at the differences between a prokaryotic cell versus a eukaryotic cell. And this particular picture, these are not to scale. The prokaryotic cell is much smaller than the eukaryotic cell, but it's enlarged just so we can take a look at the differences between the two. So if you look to the left, of course, we have the appendages that we've talked about and that nucleoid in the middle, that concentration of DNA. And then if you look on the right at the eukaryotic cells, you'll see lots of membrane-bound organelles. So it's very organized and much more complex than the prokaryotic cell. So as we said, prokaryotic cells are in the domain bacteria and the domain Archaea. So we're going to look at both of those today. And let's start out by looking at the domain bacteria. So there are a couple of things here. Their cell wall is composed of a peptidoglycan. They come in three different shapes, and they're categorized based on their growth arrangements. So here's just an image of the peptidoglycan. Most bacteria have cell walls composed of this, and they are linked, it's basically polysaccharides linked by amino acids. It's very rigid and provides structure for the bacteria. Okay, so let's look at the cell walls of these bacteria, because then we start to be able to categorize our bacteria based on whether they are what's called gram-positive or gram-negative, and this is based on their cell wall composition. So why is that even important, right? It's important because if you know what type of bacteria you're dealing with, if it's causing an infection, then you can know how to then fight it. So extremely important. Okay, so let's look at this. A gram-positive bacteria cell wall, such as the one on the left, in a gram stain, which I'm not going to go through all of that, that's in your textbook, you can read over that, but it stains as a dark purple. And So if you look here in a gram-positive cell wall, the peptidoglycan cell wall is very thick above the plasma membrane. And gram-positive bacteria are sensitive to penicillin. So if you know you have a gram-positive bacteria that's causing an infection, then a doctor would, of course, prescribe penicillin or something similar to it that could help clear up that infection. The gram-negative bacteria, on the other hand, is very thin-walled, as you see, and here's our plasma membrane. But they also have a second membrane. It's this outer lipid membrane as well. So they actually have two membranes and a thin wall, but they are resistant to penicillin. And so that's a gram-negative bacteria. So it would show up red in the in the gram stain. So this is what that looks like. Our gram-positive on the left shows up purple. Our gram-negative on the right would show up red. So that's the start to how we begin to categorize our bacteria. Another way to start categorizing the bacteria and classifying them is based on their cell shape. And in general, there are 3 different shapes. And you have seen this in the lab by now. So these should look familiar to you. The first is a spiral shape, so spirillum. The second is bacilli, which kind of look like a capsule or a rod. And the third is cocci, which is round. And these are all done in electron microscopes. So when you looked at it in the lab, you looked at 100x. They probably didn't quite look exactly like this, but if you had an electron microscope, you would see the more details like this, like we get to have here. Okay, so then now we have 2 ways of starting to classify them, and the third way is their growth pattern. So the way we say that is that if you look at this first one, and Staphylococcus, staph means that it's arranged in clusters, and remember coccus or cocci were round shaped. So based on bacteria names, you usually know a little bit about the bacteria. So Staphylococcus is cocci shaped bacteria grown in clusters, and that's what that name means. The second here, the Streptobacillus, strept means that it's a chain of bacteria. And bacillus is just what it sounds like. That would be the rod shaped. So you have a chain of rod-shaped bacteria. And then down here at the bottom we have Diplococcus. Diplo, just like you would think, means that it's pairs, 2 of them at the same time. And again, there's that coccus, which means they're round. So round pairs. And that, by the way, is usually what causes pneumonia, in case you were wondering. So that is how we tend to name the bacteria that we are looking at. So we've talked about what the bacteria looks like shape-wise, its structure. We've talked about its sexual reproduction. We've talked about sort of how we identify them, their growth patterns. So other things that we need to know about our living organisms that just help us to understand them. One is their oxygen needs for bacteria. And for bacteria, that falls into 3 different categories. One is aerobic bacteria. And this is when your Latin roots are going to start being helpful. So your word aero over here, meaning oxygen. So that means that bacteria requires oxygen to carry out cellular respiration. So that's a necessity for aerobic bacteria. And then there is facultative anaerobic bacteria. Well, let's start with the word anaerobic. An being not, aerobic, again, being oxygen. Well, facultative throws in a different sense of that, and that means that bacteria can grow with or without oxygen. So it can do either one. So it's a very flexible bacteria. Usually with those, they do have a preference, but they can function in the other environment. And then there's what's called the obligate anaerobic. Again, anaerobic being without oxygen, obligate meaning it has to be that way. So these bacteria cannot grow in the presence of oxygen. They require an anaerobic condition. Okay, what about how do they acquire their energy, right? I mean, we got to know, now we know what their oxygen requirements are. So we have two different main categories. One is our autotroph and the other is heterotroph. Troph just means feeding. So auto means self-feeder. Hetero means other feeder. So in other words, they have to acquire their food from outside of themselves. So let's focus on our autotrophs to begin with, okay? And this is kind of a flow chart to help you understand all the different categories that the bacteria can be in. Our autotrophs, to start with, we have the photoautotrophs. Photo meaning sunlight, so our photoautotrophs are our photosynthetic bacteria. Chemoautotrophs means that they're still self-feeders, and they use chemicals in order to do that. Within our photoautotrophs, they can either be anoxygenic or oxygenic, meaning that they can't, basically anaerobic and aerobic photoautotrophs. Then coming over here to our heterotrophs, they can be chemoheterotrophs, meaning they acquire those chemicals from outside of themselves, or they can be saprotrophs. Anybody know what that word means? Yeah, those are your decomposers. Saprotroph meaning that they are feeding on dead or decaying living things. Oh, well, dead or decaying organic organisms. Okay, so let's look specifically at our autotrophs, and in particular our photoautotrophs. Remember, we said there were two different categories in that, the anoxygenic and the oxygenic. And just like you would expect, the anoxygenic use anoxic photosynthesis, so no oxygen present. It does not produce oxygen because that would kill itself. And they have a specific type of chlorophyll called bacteriochlorophyll, and it's they usually live in swamps or marshes. You know, that stinky smell that would come from these bacteria, such as green sulfur bacteria. The oxygenic photoautotrophs do produce oxygen, and they have photosystems 1 and 2 just like plants do, and the example of that is cyanobacteria. So cyanobacteria, these are our oxygenic photosynthetic bacteria. They are gram-negative, and in addition to your chlorophyll 1 and 2, they also have additional pigments that can make them various colors that you see. There are 3 different growth styles. They can be single-celled, colonial, or filamentous. Cyanobacteria are usually thought to be very important because according to some theories, they were thought to have been around when the atmosphere was much different, and by being some of the first photosynthesizers, they were thought to have changed the atmosphere to include an increased amount of oxygen. Again, I think I've talked about this before, but that is one theory because it can't be tested and we weren't there. It is just a theory, but it's something that you need to know, especially if you're going into biology, because that is talked about a lot. So let's take a look at some of these cyanobacteria and what they look like, and some of these that you looked at in lab, so that it'll look a little more familiar to you now. So here is the cyanobacteria Gloeocapsa. It can live as a single-celled organism, or it can live in a colonial form, as is shown right there. They secrete a gelatinous sheath that protects the individual or the colony, so you can see that in this one right here. It really is just like a gel, and hopefully you got to see that in lab as well, but here, if you can see them. So that is one style of cyanobacteria. Okay, and here are the two filamentous cyanobacteria that we saw in lab, the oscillatoria and the anabena. And so specifically in our anabena, we had two different cell types. Let me get my little pointer here. So most of them are just vegetative cells. They go along happily producing, happily photosynthesizing and producing sugars and oxygen. And then there are these specific cells right here called heterocysts, and they perform nitrogen fixation. So that takes inorganic nitrogen and puts them into organic molecules that can be used by the cyanobacteria. Okay, bringing us back to this slide that looks familiar, we have gone with our autotrophs, our photoautotrophs. So now I want to take a look at the chemoautotroph. So the chemoautotrophs are those that carry out what's called chemosynthesis, and that's just the process of oxidizing inorganic compounds to obtain the energy needed to convert carbon dioxide into organic compounds. And usually their byproduct helps other organisms in the environment. And so they often live in a symbiotic relationship. And the example of that is the nitrogen-fixing bacteria Rhizobium. And they have a symbiotic relationship with legumes, or it's a particular type of plant. We're going to talk a lot about this when we get to plants, but just wanted to start here by introducing it because the bacteria portion of this symbiotic relationship is really important. So usually what happens is the Rhizobium live in the soil. They produce what's called nod factors, which is a chemical that induces root hairs of alfalfa in this case to start forming these nodules. See these right here? And that creates a house basically for these bacteria to live in. And the bacteria benefit because the plants provide sugars for them, and they help the plant by taking nitrites and nitrates out of the soil that are an unusable form and transforming them into ammonia, which is a usable form for the plants. I'm not going to go into the details of the heterotrophs, but just to know that those categories exist, our chemoheterotrophs obtain carbon and energy from other organisms. Those are our parasitic bacteria. And then our saprotrophs are the decomposers that we talked about that help to decompose dead or decaying material, or animal waste, for instance. Whew, okay, we've made it through our domain bacteria. Now we have the domain Archaea, which is our second domain of prokaryotes. And these usually fall into the categories of being anaerobic. They can be thermophilic, which if you remember what your Latin roots, thermo being heat, philic, they love heat. And most of them are chemoautotrophic. And there are three different types of our Archaea. A lot of times they're referred to as Archaea bacteria, so if I say that, that's why. But there are three main types. There are methanogens, halophiles, and thermoacidophiles. So let's talk about those. Oh, right. First I wanted to get to this. I totally forgot to say this. Our bacteria and eukaryotes, when you're looking at plasma membranes, have a lipid bilayer, right? We all learned that in probably middle school even. And so what that looks like is you have your phospholipid bilayer. Here you have the space in between. But some Archaea, or I would probably say even most, have a lipid monolayer. Note that there's no space in between, and that is thought to allow them to live in the extreme environments that they live in, because a lot of Archaea live in extreme environments, so they're called extremophiles. So let's talk about our methanogens to start with. They are obligate anaerobes, so that means that they require an environment that lacks oxygen, and they produce methane gas. So they live in swamps, landfills, and they live in the intestines of humans and cows. So are cows contributing to climate change? Have you heard about this? Actually, it's been one of those things that's been said off and on for a very long time. It kind of came into greater play with the new Green Deal, kind of got laughed at a little bit. Because of the methanogens that live in the gut of cows, methane is produced. So through flatulence and belching, we have the release of methane gas. So one cow produces about 200 liters of methane gas per day, which is actually a lot, isn't it? So there's your tidbit of information. So this is just another little fun fact. The methane's lifetime in the atmosphere is much shorter than carbon dioxide. But it's more efficient at trapping radiation than carbon dioxide. So that's kind of where that idea that it contributes to climate change or to global warming came about. I personally am not really convinced that a cow would be that much of a contributor, but that's just my opinion. All right, so that was kind of some fun with our methanogens. The second type is a halophile. Halophiles are aerobic, so they do require oxygen, and they are chemoautotroph. So they tend to live in very salty places, because halo meaning salt, phile means they love salt. So they live in very, very salty places. So like the Dead Sea, 7 times saltier than regular seawater, and this is where the halophiles live. The third type are the Thermoacidophiles. So just as the name suggests, they like it very hot and they like acidic conditions. So the geysers and hot springs fit that bill, and there are lots of them in Yellowstone National Park. If you've had the opportunity to go out there, you know it's beautiful, but it's also stinky because it has a lot of sulfur production. So this image right here is of the Grand Prismatic hot spring in Yellowstone, and this is looking from the top down. It's an aerial view. So just to give you an idea, this right here is a walkway, and right here are 2 people. So that just gives you the scale. Now, interestingly, in these springs and in the geysers, depending on the depth of the geyser, the deeper you go, the hotter it is, and sometimes the more acidic. So depending on what environment the Archaea bacteria desire, you can tell which ones are present, because they're what causes the color of the water. So those that can handle it really, really hot create this dark blue. As you get closer to the edges, it gets a little more greenish yellow, and that's slightly cooler, still too hot for us. But that's where the different Thermoacidophiles live, and they make this beautiful picture. So this is my youngest daughter at one of the geysers, happy as a clam to be there, and so pretty in the back. Now, what I have to show you next, this was one of my other daughters. And you like the smile? It's a fake smile. Because she hated the smell, absolutely hated the smell of these geysers because of the sulfur production. And so she would go, "Do we have to go there?" And I'd say, "You have to give me a smile for the picture." So anyway, so that's my end for this one, just to give you a little laugh. But those are the Thermoacidophiles, making one daughter happy and the other not so much. Anyway, thanks for listening today, and we will get back together soon.